Hello viewers, the topic of today's class is definition, objectives and scope of biochemistry. The purpose of the class is to enable the students to understand the importance of the science of biochemistry and also to acquire knowledge about the scope of biochemistry. It is divided into 3 modules: definition of biochemistry, objectives of the science of biochemistry and scope of biochemistry. Biochemistry basically describes in molecular terms the structures, mechanisms and chemical processes shared by all organisms and also provides organizing principles that underlie life in all its diverse forms, basically principles that are referred to collectively as the molecular logic of life. Although it provides significant insights into practical applications in various fields like medicine, agriculture, nutrition, immunology, microbiology and industry, its ultimate concern is with the wonder of life. If you look at this picture, this is from the Garden of Eden by Jan van Kessel the Younger. It includes all forms of life which contain the same biochemical macro and micro molecules. Now, with respect to the objectives of biochemistry, the first and foremost objective is to understand the high degree of chemical complexity and microscopic organization of thousands of different molecules that make up the cell's internal structures, and to understand the defined functions of each of these components, which are commonly known as nutrients and can be divided into macro and micronutrients, and the regulated interactions that occur among them. To study the systems for extracting, transforming and using energy from the environment, enabling all these organisms to build and maintain their intricate structures and to do mechanical, chemical, osmotic as well as electrical work. The fourth objective is to understand the capacity of all these living organisms, starting from the smallest bacteria to the biggest human being, to undergo precise self-replication and self-assembly, enabling a large bacterial cell placed in a single nutrient medium to give rise to a billion identical daughter cells within 24 hours. With each cell containing thousands of different molecules, some of which are extremely complex. And then to study the mechanisms for sensing, for responding to alterations in surroundings, constantly adjusting to these changes by adapting their internal chemistry with respect to the molecules. Finally, to get familiarized with the history of evolutionary change that occurs through mutations which enable the organisms to change their inherited life strategies in order to survive in newer circumstances. The third module, which contains the scope of biochemistry, defines the entire scope of the science and the understanding of the molecular basis of life. The scope can be expanded under each of the defined objectives, covering the subject under different headings. Now, these headings can be grouped under the main heading of foundations of biochemistry. Biochemistry initially has cellular foundations, going on to chemical foundations, physical foundations, genetic and evolutionary foundations. Firstly, we will deal with the cellular foundations. Basically, cells have unity and diversity of organisms which become apparent at the lowest cellular and molecular level. The smallest organisms consist of single cells which are called prokaryotes, and they are microscopic in the sense they cannot be viewed with the naked eye. Larger multicellular organisms contain many different types of cells. These are called eukaryotes. They vary in size and shape and specialized functions. Examples are plants, animals and human beings. Despite all these obvious differences, all cells of the simplest and most complex organisms share a number of fundamental principles which can be seen at the biochemical and molecular level. A detailed study of the different components of the cell and its organelles, their functions, the structure and their chemistry forms the scope of the section under cellular foundations. If you look at this diagram, it shows you the different components of the cell. This can be a plant or an animal cell containing similar features. The cell basically contains the nucleus, and this picture shows the nucleus of a eukaryotes, which is surrounded by a nuclear envelope. It is also called a nucleoid in bacteria or prokaryotes. It contains the genetic material, basically the DNA, along with its associated proteins, the histones, and also nucleus is membrane bonded. The cell membrane includes membrane proteins that function as transporters, as receptors, and also as membrane bound enzymes. Next, we have the cytoplasm, which is outside the nucleus and inside the plasma membrane. It is the aqueous watery substance in which all other cellular substances like organelles and loose molecules are embedded, and all of these contents are contained within the plasma membrane in order to maintain the diverse structure of the cell. If you centrifuge the cellular contents, you can see in the test tube shown there that the cytoplasm contains the supernatant and the precipitate contains all other organelles and cellular substances. The second picture shows you a diagram of both plant and animal cells, and this diagram clearly indicates that there are a number of components within the cell that are common to both plant and animal cells. Some of the structures are specialized plant cell structures like the chloroplasts with the thylakoid membranes which are involved in photosynthesis. Otherwise, organelles like the mitochondria, the endoplasmic reticulum, the lysosomes, the peroxisomes which destroy peroxides and other free radicals that enter the cell, the cytoplasm, the nucleus, the nucleolus, all these are common to both plant and animal cells. In addition, plant cells have a huge vacuole which contains water, and this vacuole helps to maintain the integrity and shape of the plant cell. Another main distinguishing feature of the plant cell is the presence of the plant cell wall, which contains cellulose, hemicellulose and other polysaccharides or other complex polysaccharides which give rigidity to the plant cell. Now, if you look at the cell and its components in this picture, you will find that there are 4 levels of organization. In the first level, you have individual macromolecules like nucleotides, amino acids or sugars. Sugars are called monosaccharides, amino acids are known as amino acids, and nucleotides make up DNA or RNA. Now, this DNA and RNA is the second level of organization of the macromolecules, and the third level includes supramolecular complexes like the organization of DNA and RNA into chromosomes, the organization of proteins into the plasma membrane, and the organization of individual glucose molecules into cellulose microfibrils which form the cell wall. All these level 3 structures organize themselves into the level 4, which is basically the cell and all its organelles. Biochemistry aims to explain the biological form and the function in chemical terms. One of the most fruitful approaches to understand biological phenomena has been to purify an individual chemical component, such as a protein, from a living organism and to characterize its structural and chemical characteristics. By the late 18th century, certain chemists had concluded that the composition of living matter is strikingly different from that of the inanimate world or the organic world. Antoine Lavoisier from 1743 to 1794 noted that relative chemical simplicity of the mineral world or the inorganic world and contrasted it with the complexity of the plant and animal world. The latter he knew were composed of compounds which are rich in elements like carbon, oxygen, nitrogen, hydrogen. During the first half of the 20th century, parallel biochemical investigations of glucose breakdown in yeast and in animal cells revealed remarkable chemical similarities in these two very different cell types. Later, this pathway or biochemical mechanism was known as glycolysis. The breakdown of glucose in yeast and muscle cells involved the same 10 chemical intermediates, the same enzymes through evolution. Subsequent studies and many other biochemical processes in many different organisms have confirmed a generation of the observation neatly summarized by Jacques Monod as what is true of E. coli is true of the largest elephant. So, the current understanding that all organisms share a common evolutionary origin is based in part on this observed universality of chemical intermediates and transformations. Only about 30 of the more than 90 naturally occurring chemical elements are known now to be essential to organisms. The four most important and abundant elements in living organisms in terms of percentage of number of atoms are hydrogen, oxygen, nitrogen and carbon, which together make up of more than 99% of the mass of most of the cells. They are the lightest elements capable of forming 1, 2, 3 or 4 bonds respectively. In general, the lightest elements form the strongest bonds. The trace elements represent a minuscule fraction of the weight of the human body, from 1 to 4%, but all are essential to life, usually because they are essential to the function of specific enzymes, proteins, hormones, etc. The oxygen transporting capacity of the hemoglobin molecule is a classic example of the need for the element iron, which makes up 0.3% of its mass. Now, if you look at the structure of the carbon, it has 4 valences. It is classically called the tetrahedral carbon atom because the 4 bonds can attach themselves to 4 different chemical groups. When 2 carbons are attached together, 3 bonds are free and 2 bonds form the link between both the carbon atoms. If it is a single bond, as the black arrow indicates in section B, it has more flexibility around the bond. If it is a double bond, as in section C, the flexibility is limited and it makes the carbon-carbon bond more rigid. These are mirror images of the tetrahedral carbon atoms. When a carbon atom is asymmetric, it is called a chiral molecule, and here the rotated molecule cannot be superimposed on the mirror image as shown in section A. In section B, we can see an achiral molecule which can be rotated and superimposed on its mirror image. Hence, the scope of biochemistry in terms of its chemical foundations deals with the chemistry, structure and relationship of structure with function and deals with the function in terms of the changes in the structure of the various macro and micromolecules that make up the cell and participate in all biological functions. where living organisms must perform work to stay alive and reproduce themselves. And the synthetic reactions that occur within the cells, like the synthetic processes in any factory, require the input of energy. Energy is also consumed in the motion of the bacterium or an Olympic sprinter. Both can be physically similar in the flashing of a firefly or an electric discharge of an eel. The storage and expression of information requires energy, without which the structures rich in information inevitably become disordered, disoriented and meaningless. In the course of evolution, cells have developed highly efficient mechanisms for coupling the energy obtained from sunlight or from the fuels, as in plants or animals, to the many energy consuming processes that they must carry out. Cellular energy conversions can be considered like all other energy conversions in the context of the law of thermodynamics, which have physical foundations. A living organism is an open system. It exchanges both matter and energy with its surroundings. They derive the energy from the surroundings in two ways. They take up the chemicals or the fuels like glucose from the environment in the form of food like we do, and extract energy by oxidizing them, and they absorb energy from the sunlight like plants. The first law of thermodynamics, developed from physics and chemistry, but also fully valid for biological systems as well, describes the principle of the conservation of energy, meaning in any physical or chemical change, the total amount of energy in the universe remains constant, although the form of energy may vary. And cells are consummate transduters of energy, capable of interconverting chemical, electromagnetic, mechanical and osmotic energy with great efficiency. A separate branch of biochemistry like biophysics has emerged to enable a detailed study of all these processes. This picture shows that the potential energy that a living system derives from the environment through food, nutrients or sunlight is transduced to accomplish work through the process of chemical transformation within the cell, and it gives out heat into the environment, and there is increased randomness, which is called entropy of the surroundings, and decreased randomness, which is called entropy in the system. So, through these processes, energy from the environment is converted into chemical energy and heat energy under the theory of biophysics. Now, we go on to the genetic foundations, which is the most remarkable property of all living organisms, because they have the ability to reproduce themselves for countless generations with near perfect fidelity. This continuity of inherited traits implies constancy over millions of years in the structure of the molecules that contain the genetic information. And very few historical records of civilization, even those which are etched in copper or carved in stone, have survived the number of years that this genetic information has survived. And many bacteria have nearly the same size, shape and internal structure, and contain the same kinds of precursor molecules and enzymes as bacteria that lived nearly 4 billion years ago. Among the seminal discoveries in biology in the 20th century were the chemical nature and the three-dimensional structure of the genetic material, that is DNA or deoxyribonucleic acids. This DNA consists of individual nucleotides in the linear polymer shape, which encodes the instructions and provides a template for the production of identical DNA molecules to be distributed through progeny. And the continued existence of biological species requires its genetic information to be maintained in a stable form. Effective storage, expression and reproduction of the genetic message defines individual species. distinguishes them from one another, and assures their continuity over successive generations. You look at the mother zebra and the daughter zebra, and you see how biological reproduction occurs with near perfect fidelity. And you look at the picture of DNA here, you find DNA is a double stranded helix with the pink, yellow, gray and orange colors depicting the 4 different nucleotide bases: adenine, guanine, thymine and cytosine. Adenine always pairs with thymine through 2 bonds, guanine always pairs with cytosine through 3 bonds, and these bonds are nothing but non-covalent bonds. But between the A, T, G and C, you have the covalent phosphodiester bonds. When the DNA divides into the progeny or the daughter cells, each strand replicates itself to form identical copies of the DNA. And if you look at the genes, genes are nothing but small portions of DNA which are called the exons or the coding DNA, and the introns in between is the non-coding DNA. So, now this DNA transcribes its sequence into a messenger RNA, which translates again into a protein. This protein starts growing on the ribosome, as you can see the pink, green and blue protein structures evolving from the ribosomal structure to form a fully native 3-dimensional quaternary structure shown as protein 1, protein 2 and protein 3. An assembly of these 3 into one unit is called the formation of a supramolecular protein complex, which is the quaternary structure. Now, we come to the last portion of today's class, that is the evolutionary foundations. Nothing in biology makes sense except in the light of evolution. This was quoted by Theodosius Dobzhansky, an American biology teacher, in as late as March 1973. Great progress in biochemistry and molecular biology during the decades since Dobzhansky made this striking generalization has amply confirmed the validity of the evolutionary theory. The remarkable similarity of metabolic pathways and gene sequences in organisms across the phyla argues strongly that modern organisms share a common evolutionary progenitor and were derived from it by a small series of changes which are now known as mutations. Each of these mutations conferred a selective advantage to some organism in some ecological niche. Despite the near perfect fidelity of genetic replication, infrequent and unrepaired mistakes do take place in DNA replication processes, leading to changes in the nucleotide sequences, producing what is called a genetic mutation. This mutation can be harmful if it leads to cancer, or it can be handed down to the offspring in the reproductive cells, which are harmful or lethal to the organism. They may, for example, cause the synthesis of a defective enzyme that is not capable of catalyzing an essential metabolic reaction. On the other hand, a mutation better equips an organism or cell to survive in the changing environment. The mutant enzyme might have acquired a slightly different specificity that is making it now able to use some of the compound that the cell was not earlier able to use and metabolize. If a population of cells were to find themselves in an environment where that compound was the only source of energy or fuel, then the mutant cell will be able to better utilize it than the unmutated or what is called as the wild type cells in the population. The mutant cell and its progeny would survive and prosper in the new environment, unlike the wild type cells. This is what Darwin meant as what you call the survival of the fittest under selective pressure. So, the scope of biochemistry finally deals effectively with the chemistry of mutations and their consequences to the living organism. Recent science has been seeing evolution of a new branch of biochemistry, more commonly known as recombinant DNA technology, which is being exploited for manipulation and production of species having better traits, which can also eliminate undesirable traits spanning across varied living organisms. Mutations can occur from time. If you see the DNA structure TGA-GCTA, the two daughter DNAs have acquired a single A mutation. One of the daughter strands has got the mutation, the other has not. And when that particular strand again replicates, the A is carried over along with another mutation called G. So, like that the mutations can pass on, adding further mutations which help in survival. So, to conclude, the basic cellular, chemical, physical, genetic and evolutionary foundations consist of the scope of biochemistry, and an understanding of all these foundations has shown us that landmarks in the evolution of life on earth, like what is seen in this picture, has caused diversification of multicellular eukaryotes. If you look at millions of years ago, from 500 million to 1000 million to 4500 million, where the foundation of earth was discovered. Dear viewers, I hope you understand today's class on biochemistry. To summarize, diverse living organisms share the same common chemical features, the same macromolecules, the same pathways of synthesis and catabolism, and derive energy from the same sources from the environment. Let us meet in the next session and continue these classes on biochemistry. Thank you.